Hello everyone and welcome back to Bioblinkers. In today's video, I'll talk about the process translation. So you people already know that when the information is transferred from DNA to RNA, this process is known as transcription. And when the information is transferred from RNA to protein, this process is known as translation. So before I proceed with the translation process in detail, let me discuss the few points regarding the mRNA. First, that mRNA synthesis always takes place in 5' to 3' direction. And second, there is a presence of uracil instead of thymine. And third most important point, that 3 nucleotides of the mRNA constitute one codon. And each codon codes for a specific amino acid. So in a genetic code, total there are 64 codons, and out of 64 codons, 3 are stop codons. That means rest 61 codon codes for a specific amino acid. So in a translation process, there is a hero, and here the hero is the ribosome. So ribosome is a non-membrane bound organelle. That means it does not have a membrane. It is made up of rRNA and protein, and it consists of two subunits, the large subunit and small subunit. The ribosome in case of prokaryotes is 70S, and in case of eukaryotes it is 80S. Now the important thing which you have to remember, that the ribosome, it has a 3 binding site, A site, P site and E site. A site is the amino acyl tRNA binding site, P site is the peptidal tRNA binding site, and E site is the exit site. The function of these sites will become more clear when we'll discuss the translation process in detail. Now let's come to the structure of tRNA. So tRNA, it has 4 things, the acceptor arm, sil loop, D loop and anticodon loop. So the important thing which you have to remember here, that the sequence of the anticodon loop is complementary to the codon of the mRNA. Let's say the codon here is UCG, and UCG code for the serine amino acid. So the tRNA containing the serine amino acid will come and bind to the codon UCG. That means that there is a specific tRNA for the specific amino acid. Now let's begin with the translation process. The first step in the translation process is the charging of tRNA. The enzyme involved in this process is amino acyl tRNA synthetase, and this enzyme has 3 binding sites, ATP binding site, amino acid binding site and tRNA binding site. Now when the ATP and amino acid binds, they form a complex which is known as amino acid AMP complex, and 2 phosphate groups are removed. Once this complex is formed, now the tRNA will come and bind to the tRNA binding site of this enzyme. After that, this AMP is released and the amino acid get attached to the tRNA. Now the tRNA is known as the charged tRNA, and the whole process is known as the charging of tRNA. Now to initiate the translation process, 2 things is required. Number 1 is ribosome binding site and second is the initiation codon. Now you people know that ribosome has 2 subunits, the large subunit and small subunit. And small subunit of ribosome is the one which will bind to the ribosome binding site. Once it binds to the ribosome binding site, now the small subunit of ribosome will start stalling over the mRNA until it encounters the initiation codon. Once it finds the initiation codon, now initiation factors play a very important role. There are 3 initiation factors, IF1, IF2 and IF3. IF1 will bind to the A site, because of which the initiator tRNA cannot bind to the A site. IF3 will bind to the E site, because of which the association of the large and small subunit of ribosome does not take place. And IF2 help in the recruitment of the charged tRNA to the P site where the initiation codon is present. Now when this initiator tRNA binds to the P site, after that the IF2 get released. After that IF1 get released, and after that IF3 get released. When this IF3 get released, the large subunit of ribosome will bind to the small subunit and result in the activation of the ribosome. Now let's say in the A site, AGU is present. Now AGU will code for another specific amino acid. So the tRNA containing the specific amino acid, which is coded by AGU, will come and bind to the A site. And when the two charged tRNA come in close proximity of each other, they result in the formation of the peptide bond. So the peptide bond is formed between these two amino acids. Now, as the polypeptide bond is formed between the respective amino acid, the ribosome will move forward. As the ribosome will move forward, the tRNA along with their codon in the P site will move to E site, and the tRNA along with their codon present in the A site will move to P site. So the A site will become vacant, and this vacant A site will be occupied by the next tRNA along with their respective amino acid. Now the peptide bond will form between the respective amino acid. Now, as the ribosome will keep on moving forward, the tRNA present in the E site will be released and the A site again will become vacant. Now, let's say this time the codon present in the A site is a stop codon. And you people know that there is no specific tRNA for the stop codon. So, stop codon will be recognized by the release factor. So, release factor will come and bind to the stop codon. And once it binds to the stop codon, the polypeptide chain gets released and the translation gets stopped. Thank you for watching this video. If you enjoyed this video, don't forget to subscribe to my channel and hit the bell icon so you don't miss any notification. Thank you and see you in the next video.